In this review, we're going to look at DNA and gene expression. So remember, DNA codes for proteins. It's made of a chain of nucleotides. It's double-stranded, forms a double helix, and the two strands are held together by hydrogen bonds. So here we have our nucleotide with its sugar, phosphate, and base. And they're arranged double-stranded. And so the sugar and the phosphate makes up the sugar-phosphate backbone, which is the structural part of the DNA molecule. And then the bases are in the center, and they're held together by hydrogen bonds. Remember the complementary base pairing rule? So A goes to T in DNA, and A goes to U in RNA, and then C always goes to G. This is not a bad thing to write down first thing on the EOC. And now this code is set up as codons, or sets of 3 bases. If I ask you to do the complementary DNA strand, all you'll do is you use the complementary base pairing rule. And so C goes to G, G goes to C, and T goes to A. And you'll do that for each one of the codons for the second complementary strand. Now moving on to protein synthesis. Remember, DNA to RNA, that's transcription. We're making a copy. We're transcribing the DNA to RNA. Then the RNA is going to be made into a protein in the process of translation. So we're going from the language of nucleic acids to amino acids. Another way to simply remember this is the C comes before the L. So first let's look at transcription. And in transcription, we're going to take our DNA molecule, we're going to use an enzyme, and we're going to transcribe a copy of the DNA into mRNA. Now, because we're using mRNA, A is going to go to U. So if this is my DNA, when I go through transcription, the mRNA is going to be T goes to A, G goes to C, A goes to U. And I'm going to do that exact same thing for each codon. Now with translation, remember the analogy. Basically what we're doing here is we're going to take materials, use trucks to take them to the factory to put together our product. But in this case, the materials are amino acids, the trucks are tRNAs, so transfer RNAs, and the factory is a ribosome. And so we're going to put the amino acids together to make a protein. So here we have our more realistic image. So we have our mRNA providing the code, the ribosome that's actually putting the amino acids together. We have our amino acids being carried by our tRNAs to make our protein. So remember, we're going to be using this codon chart. And basically what this codon chart is doing is it's taking mRNA codons. Notice they have U's. So if you're given DNA, you have to transcribe it first. And it's translating them into amino acids. So AUG goes to methionine, and so on, all the way to a stop codon. So if we start out at the beginning, we have our DNA. We have to go through transcription first. So now we make our mRNA, and we've used U's. Then we're going to go through translation. We're going to use our chart, and we're going to translate those mRNA codons into amino acids. And this gives us our protein. Next, we're going to look at environmental influences. So remember DNA protein trait, and how the genes or the DNA becomes a trait is what's called gene expression. And environmental factors can have a major impact on this. And we talked about a couple different ways. Now, these can be beneficial because they allow an organism to respond to changes in their environment, but they can also be detrimental in that they can cause many different diseases. So here's an example of what we call gene activation. So when the temperature outside is warm, these Himalayan rabbits will be white because the pigment gene has been inactivated, so it doesn't work. When it gets cold though, that gene will turn on or be activated, and you will get a change in phenotype. No change in the DNA, just a change in whether or not the gene is activated. Another example is genetic predisposition. So we have two twins and then a third individual. One of the twins in that individual is a smoker, but only person number 2 gets cancer. Why is that? Well, that's because the twins were actually genetically predisposed. In other words, they were more likely to get cancer because of their DNA. However, twin number 1 did not get cancer because the gene was not activated because the environmental factor was not present. Person number 3 was actually genetically protected against cancer. We also talked about how this can affect the amount of protein that is created. So when the sun hits your skin, it's going to cause more protein to be made, and your skin is going to get darker to protect you. Finally, we looked at these plants called hydrangeas. The exact same plant is going to be blue or pink depending on the pH of the soil. So this environment actually changes how this gene shows up. All right, cell differentiation. Remember, all your cells have the same DNA. So how do we get all of these different cells that make up our body? Well, they turn on and off different genes. So for example, a blood cell would turn on the hemoglobin gene, whereas a bone cell would not. And this is what makes them different, using different genes. And this is the process called cell differentiation. And finally, we're going to look at mutations. So remember, a mutation is a change in the DNA code. And these can be additions or subtractions or substitutions. And remember, an addition or subtraction generally worse than a substitution. Now, if you change the DNA, you're also going to change the RNA, which means you're also going to potentially change the protein and also potentially the trait. Now, most mutations, remember, are detrimental or neutral, but some are beneficial and can therefore be acted on by natural selection through the process of evolution.